So welcome everybody here in the room and online. Today we have our cluster seminar, and it's a great pleasure for me to introduce Valerie, who will present the SoBee project. Let me tell you 2 or 3 things about Valerie. She has done a PhD across the world. She worked with Adrian Dyer in Melbourne in Australia. She worked with Martin Jurfa and people in the group in Toulouse in France. She worked in Würzburg. So she has moved across the world. And the basic question was, if you look at collective behavior, a honeybee hive, there's so many animals together that work, but what does the individual do? How much individuality is there in a hive? How different is one animal from the next one? And how consistent is that? Some people today, in a fancy way, call it personality, but maybe just how consistent are the properties that an animal, a single honeybee, performs in its behavior. So she looked at learning and memory at an individual basis over time in a hive, and published in her PhD. And then we were super proud when last March she said, I will come to Constance. I will come to the cluster. I will look at honeybees in this context, and specifically I will look at how do bees keep their hygiene. What do they do as a collective to treat diseases within the hive? As I said, Valerie joined us last March, so she has been here now for a few months, already building up many things. But my understanding of what you want to do today is to present the plans of this research and to get some feedback and to expose what do we do, what will Valerie do, and what are the best experimental and conceptual approaches to studying this question. And I would like to give you the word, and I'm really looking forward to your talk. Thank you.
Thanks, Giovanni, for the nice introduction, and thank you all for being here in person, and also big thanks for the people on Zoom. And yeah, as Giovanni said, today I'm going to give more of an introductory talk into my topic, which is the SoBee project, Unraveling the olfactory neuroethology of hygienic behavior in honeybees. Here, I want to directly approach you guys and ask a question to the audience. So did you ever feel like this? So I for sure did. And what is then going on basically within my body is most probably an innate immune response happening. And we have specialized cells within our body, so-called macrophages here, which patrol our body and are looking for pathogens and that don't really belong inside our body. So what happens when a nasty pathogen enters our body? Once the macrophage will come into closer contact with the pathogen, via chemotaxis, it will recognize the pathogen as not belonging to our body, basically saying, you smell funny, you should be erased. And then the macrophage will eventually get rid of the pathogen. And this is what I said, the innate immunity, where we have innate reactions to diseases. However, in humans, we also have an adaptive immunity, which helps us to cope better with diseases that we have been exposed to. However, honeybees rely more on an innate immunity and have lost most of their genes related to adaptive immunity. And I want to draw this picture and actually want to say, diseases also affect bees just like they affect us, however, in a different way. So as you have seen, the macrophage patrols the body and is responsible to get rid of the pathogens. In our eusocial honeybee colony, it's basically the same, but here, individuals are basically the cells of the eusocial organism and perform exactly the task that the macrophages does. So really, diseases affect everyone, and it's important to unravel the mechanisms which drive these behaviors. Now, because we have a wider audience, I want to talk a little bit about the life cycle of honeybees. and especially want to highlight that the collective is key. So honeybees have a eusocial lifestyle where they live together with more than 10,000 individuals within a single hive. They have a division of labor into reproductive and non-reproductive individuals. So here we have the queen, which is the only female, fertile female in the hive. We have the males, which are reproductive, and all female worker bees are non-fertile. We have overlapping generations and cooperative brood care. So here, this really furry bee just emerged probably that day I took the photo, while this bee is older, and at all times there are multiple generations within a single hive. And also the queen lays the eggs, and then after 22 days, the bees emerge, and the already emerged bees take care of the brood and divide their tasks. And this is what is done here. So we also have an age-dependent task allocation among the bees, which looks a little bit like this, where we have the life cycle of the bee. On day 1, the queen lays the egg. 22 days afterwards, the bee hatches. It first starts with cleaning duties, then becomes a nurse bee, building, guards, and then onwards until the bees die. They will perform mostly foraging tasks. However, this is rather a simplified scheme, and it's more complex than it seems. However, the downside of living as collective are pathogens and parasites, because they offer optimal condition for the transition of diseases. This is because to the extremely high density of individuals, so social contact is really necessary for the individuals. Their whole colony structure is built about contact, social contact among individuals. And finally, the group members within a hive are all relatives. However, honeybees within the last years have been collapsing, and pathogens play a significant role. A major parasite that causes honeybee diseases is the ectoparasitic mite Varroa destructor, which life cycle is tightly linked to the honeybee biology. So the Varroa mite cannot live without its host. And what the mite basically does, the female mother mite goes into the open cells and will consume the hemolymph of the larvae and will reproduce, males and females. And then once the bee hatches, the whole mite family goes out and can disperse further into the colony. And this loss of hemolymph during the ontogenetic development is associated with a decrease in weight, earlier foraging onset, and decreased lifespan, and also decreased learning, longer absence from the colony, less returning. So the bees are really just less healthy. And also the mite can carry viruses such as the deformed wing virus, which does what it sounds like. It leads to crippled wings. And these individuals are often thrown out of the colony because they are not healthy. And even if they might function when they are young, once it comes to foraging, this bee is really useless for the colony. So it's a high impact. Furthermore, we have the American foul brood and the European foul brood bacteria. They are brood diseases and basically turn the whole brood. There should be nice white larvae in here. However, this is a brown goo. And the treatment for this is quite drastic because you have to burn all of your colonies. You have to inform the veterinary public government veterinary things because it can spread. There's no cure, while Varroa can be treated with different kinds of acids and is kind of under control. But there's really no control. However, as always, there's a solution to the problem. And the solution of the collective is what we call social immunity. So as I said, instead of focusing on an adaptive immunity system, the honeybees have evolved collective rather than individual immune defenses, which can range from constitutive, so mechanisms that are always there, and inducible behaviors that the bee performs once a disease spreads within a colony. One constitutive mechanism is the polyandry, so multiple matings of the queen to produce genetic diversity within a hive, so that not all members within a colony are full sisters, so have the same mother and the same father. Furthermore, we have task allocation. We have the transfer of compounds and microbiotica that help to fight diseases. We have the use of resin, which also has microbial compounds. We have the constant allogrooming of all individuals within the hive. And here we come more to the inducer mechanisms, which are, for example, social fever, where the collective raises their body temperature by a few degrees, which also kills pathogens. And finally, we have the hygienic behavior, which is the opening and removal of diseased brood cells. And in the SoBee project, we especially focus on the hygienic behavior. So let's dig a little bit more into the hygienic behavior. Here we see a nice sketch. Here we see how a normal sealed brood comb looks like. So once the larvae of the honeybees go into pupation, the wax combs are sealed because the pupae don't need any food, so they are sealed. And here we see an intact wax cap. Then if a hygienic bee senses something funny, it will eventually open the cell to investigate it, uncap it, so remove the cell cap. And then two things can happen. If the bee thinks, oh, something was funny, but now that I opened the cell, it looks quite good, it could recap the cell, and then I hope you can appreciate that here, there's a round circle that we don't have here, so you can see that the cell has been recapped. Or if they think that the pupa smells funny, or as you can see, there's a nasty varroa, they get rid of the brood and will remove it. So then there's the question, because we have seen there's an age-dependent division of labor, who does it and which signals trigger this behavior? And this is really not so well known throughout literature. So as a first experiment, because I wanted to have a look at what the behavior actually looks like, I took a brood comb like this. I marked with a blue paint, I marked cells, and then I damaged the cells with a needle and killed the pupa inside. And then for several hours, I recorded zoomed in what the bees were doing upon this. And here you can see a video. This was taken just as I did the manipulation. I hope here you see the frame where inside all the cells were peaked. Here you see this jiggling of the head, which indicates the uncapping of the sealed brood. Here we see that the bee is licking the cell contents of the open cell. And here you see a semi-opened cell. And this is what the behavior looks like. And you see that it's really quite messy, and you cannot identify one bee that is doing everything, but it's really more a collective behavior that a bee comes, uncaps a little bit, then leaves it again, then comes back. And basically this goes on. Is this? Yes, and this is a video after a few hours. And now you see here the cell is fully opened, and the bee is eating the pupa in the open cell. This is also happening here. Yes, this is also happening here, and we can see even several hours later, some cells are opened. However, some cells here are still closed. And we really see that it's a collective behavior, not one bee doing all the work. And it's not so easy to identify the bees that are actually performing the hygienic behavior. So when we now come back to who are the bees that are actually performing the hygienic behavior, there's a nice experiment done by Bienefeld et al., where they use this hive setting, but basically we only care about J, where a brood comb was filmed, and it was one brood frame like this, and the authors introduced 2,000 marked bees into this J here. Together with the brood comb, they infested 29 cells, brood cells here, with varroa. They tracked the uncapping behavior with a camera. And what the authors found, that out of the 2,000 bees that the author put into this setup, only 16 bees were involved in the uncapping behavior. So it's only a small subset of the bee population. And the bees are involved in multiple uncappings and open multiple cells. Then there was the question, how do the bees sense that something within the sealed cap is not normal? And soon the hypothesis arose that it might be olfactory signals. This was analyzed in a study from McAfee, where they put freeze-killed brood, so brood killed by putting them in liquid nitrogen, versus live pupae. And here in the first set of experiments, they did a solvent extraction for the cuticular hydrocarbon profiles, so the non-volatile compounds that the bees have all over their body. And in the second set of experiments, they compared from 6 hives, here again the cuticular wasps, and they also did a SoBee extraction, so more of the volatile compounds that are more volatile. And this is the chemicals they identified in the different stages. And when they analyzed this, here we see the compounds that they actually found that differed most between live in green and in black the dead pupae. And one component that was significantly increased in the dead pupae was oleic acid for the cuticular wash, and for the volatile extraction, it was especially beta-oxymene that was significantly increased in the dead compared to the live larvae. And then they tested whether the odorants would induce hygienic behavior in honeybees. So what they did, again, they took a brood comb and put the oleic acid, beta-oxymene, the negative control hexane, because the odorants were solved in hexane, and phenetyl acetate into the hive. And then they looked into how many cells were uncapped. This figure looks a little bit complex, but I will move you through it step by step. So here in A, we see that the authors tested 6 colonies that have a high hygienic score, so removed almost all killed pupae after 24 hours, and contrasted them with a low hygienic score. So basically, this is what we see here. They used a 1% odorants and 100% odorants. And here we see the different odors. And I hope you can appreciate that with beta-oxymene and especially oleic acid and the mix, the rate of removal is significantly increased, especially in the individuals that have a high, in the colonies that have a high hygienic score. So indeed, it seems that the hygienic behavior is triggered by olfactory signals, and here in this example, for especially oleic acid. Okay, that was nice. So hygienic behavior seems to be triggered by olfactory signals. However, it's not that simple, because then I did a literature research and was looking into odors that might trigger hygienic behavior. And I found quite a lot of odors that are dependent on different diseases, whether it's bacteria, whether it's parasites. And I ended up with a huge list of possible odors that trigger hygienic behavior. So it's really not that simple that one odor is a universal marker. At least it does not seem like it. So this will be fun to find out which of these signals are most prominent. And now let's come to the SoBee project and to the project overview, which we can see here visually. And I hope I will move you through every step. It's a little bit overwhelming, but I hope you appreciate that we have 3 work plans that we divide by 3 universities. Here we have us, University of Constance. We focus on the behavioral experiments, on social immunity, gland dissection, and data generation using calcium imaging. Our partners from DTU will concentrate on yeast biotechnology, and our third partner is a chemical expert. So let's talk first about the Uni Constance work plan. So we have expertise in honeybee behavior and neurobiology. We will focus, as I said, on behavioral experiments, which are mainly odor preference assays and cleaning assays using different setups, which I will talk about in just a few minutes. We will dissect honeybee glands, and the ultimate goal is to perform calcium imaging to characterize the glomerular activity patterns to different disease odors. Then I want to introduce our partners, first from the Technical University of Denmark, Dr. Emil Damgaard Jensen and Dr. Akriti Soni from the Microbes Initiative. They are experts in yeast biotechnology and synthetic biology. Their work package includes the deophanization of honeybee olfactory receptors in yeast, the development of a deophanization platform to screen olfactory receptors of the honeybees to identify compounds that are involved in hygienic behavior, and the development of a yeast-based odor map. Our other partner, Dr. Oliver Fien from the Metabolomics Lab at UC Davis, has expertise in analytical chemistry. His work package includes the identification and characterization of volatile organic compounds and gland extracts that we from the University of Constance prepare. He will do volatile metabolomics using GC-TOF MS. And finally, they will refine and standardize volatile biomarker assays for volatile profiling of the bee glands and pathogens. And now let's start where I moved in, where I jumped right in March and getting the behavioral setups established and starting with the odor preference assays. And here first, I want to start with explaining the whole reasoning behind it. So as you've seen, the hygienic bees are quite rare within the hive. It's only a small subset of the colony. And there's only one paper that has really looked into the ethology of those bees. And they state that hygienic bees are middle-aged bees between the age of 15 to 17 days, so considered old nurses, but still before they start foraging. So it's basically, they are too old for being nurses, but they are still too young for being full foragers. And if this is the case, our hypothesis is that bees show an age and task, because age and task are always related, dependent sensitivity to task-associated odors. So what do I mean by this? Basically, we have 3 groups of bees. We have the young nurses, we have the supposed to be hygienic bees, and the forager bees. And if the hypothesis, we would expect that the nurse bees should have an innate odor preferences for the task that they are performing, so for brood odors. Our hygienic bees should thus prefer or have an innate preference for disease odors, while the forager bees should show a preference for floral odors because it's their job to forage for resources. How I started with this was using the YRPS setup, which is a fully controlled odor delivery system where we have a flow meter which go through solenoid valves and into the setup. So this is the YRP setup. The odor goes in here, a cloth cover, and here are the odor vials, and through shunting the sonoiloid vials we have either clear airflow or the air goes through the odor which is stored in the vials. And we have a vacuum pump that removes all the odors out of the setup. And it's controlled fully automatic with a computer, so you can program the odor delivery, the intertrial interval, and everything. And how this basically looks is now you mostly see nothing. Maybe you see the bee walking inside here. However, we don't need to see it because we can track the bee's position always on the computer with light barriers. And this is the last protocol I used for the bees in order to get odor preferences. So this is a sketch of the YRPS setup. I blocked one arm and had to make a dual choice arena. I introduced the bees and gave them 5 minutes of darkness to habituate to the setup. Then I turned the UV light on in the middle for 10 seconds because this was a great tool to lure the bee into the middle. And then I introduced either an aversive odor, butyric anhydride, smells like rotten butter. That's why we have this. And on the other side, we have an appetitive floral odorant for 30 seconds. And I repeated odor delivery 9 times. And I tested the same individuals for 2 days. And this is basically what the setup looked like. And now you will see the UV light turning on, and then the bee, it's almost like a barrier. The bee will always stay inside this UV circle. And once the UV light turned off, the odorants were delivered here and here. Now let's come to the results. So is the setup suitable to test odor preferences? Here we see the example of one individual. Here we see 1, 2, and 3, nothing happening in 3 because it was blocked. The black lines indicate the path of the bee. And here, this indicates the odor stimulation in arm 1 and in arm 2, always for 30 seconds. And I hope here is the speed of the bee, and I hope you can see nicely then whenever the UV light was turned on, the bee was really in the middle and not moving. And here we see for this individual bee, her preferences. So this bee spent more time in the butyric anhydride arm compared to the linalool arm. And this was also true when we had a look at the group performance. So this is for day 1. And the bees showed a significant preference for the aversive odor. However, when I tested the same individuals on the 2nd day, this preference was completely vanished. And also when I had a look at the individual performances, so here is the percentage of time spent in the environment on day 1 and day 2, we see the blue circles represent the data of 1B. We see that there's no correlation, and the same is true for linalool. This, for me, really shows that in the YRP setup, there's no odor preferences because the odor preference should be stable in time. If I prefer linalool on day 1, I should also prefer it on day 2, and this is really, I could not observe this. So is the setup suitable to test odor preferences? After spending months and after 35 protocols, the answer is maybe yes. I never managed to at least reliably get it done. So it was really time to focus on another setup. However, the troubleshooting of the YRPs is still ongoing with the help of Hannah Schnell, and ideas are always appreciated why I cannot get the bees to get odor preferences. Then we had the TK practical, which was now perfect for me to find alternative setups that I could use. Here I tested the arena setup, where now we scented the bees inside an arena and observed the behavior of unscented and sun-scented individuals. And we had the observation hive setup where we have a whole colony here, nice on glass, so we can observe what the bees are doing. And also here we introduce a scented individual and observe the behaviors towards this individual. Here a huge thanks goes to Lea Bartel and Natalie Kaiser for helping with the experiments. So this is how the arena set up basically looked like. So bees were caught, returning foragers from the hive entrance were caught, they were chilled on ice, then a colored dot was applied, the scented individual was always marked in pink, and the unscented individuals in blue. We used 1 microliter of either limonene, a floral appetitive odor, oleic acid, death pheromone, and the alarm pheromone isoamyl acetate. And this is basically what it looked like. So we put the bees into an arena and then observed the behavior for 20 minutes and performed 7 repetitions per odor. And we scored a different set of behaviors that we have observed in preliminary experiments. And here we are for the results. First, for the floral odor limonene. Here we are contrasting the behaviors of the scented individual in black and the gray individuals, the unscented individuals, and we compare the different behaviors and how the percentage of time the behavior was performed differed. And we see that for limonene, unscented and scented individuals did not behave differently. The same was true for the alarm pheromone and the oleic acid. Okay, nice. So in this setup, in this semi-social setup, scented and unscented bees do not behave differently. However, when we have a look at the behaviors, we see significant differences. So here, this is for the overall interaction, comparing the floral odor, the alarm pheromone, and the death pheromone, and we see that the overall interaction of the oleic acid scented bee is significantly reduced, while the proboscis extensions are significantly increased when the bees were scented with oleic acid. So scented and unscented bees behave differently in response to the different odors I tested. So is the setup suitable to test odor preferences towards differently scented bees? Yes, these are first promising preliminary results. However, the protocol needs to be adjusted. So manual observation is not so reliable. So right now I am working on a deep lab cut protocol to have a precise pose estimation. I will even the number of scented and unscented individuals 2 and 2, because before there was just 1 scented individual. And I'm currently redoing the experiments, and they are now almost done. I think it's 4 videos left to get a first glance at the data. And an update on this follows in a few slides, because now I also want to talk about the observation hive setup, where the protocol was actually really similar. So we received a bee from the observation hive, we removed the glass, got a bee in the syringe, cooled the bee. Now we applied a number tag, because lots of colored bees in the hive, then it's easy to lose your bee. So we had an individual number tag, and now we also introduced a pentane control, which we didn't have before. Then the prepared bee was introduced exactly on the frame where we had got her from. And then again, the behavior was observed for 20 minutes, and 7 repetitions were done per odor. And here we see the results. It's the same graph as in the previous experiment, so contrasting the different behaviors across the treated bee and the resident bees, which are inside the hive. And now for the floral odor limonene, we see that the treated and resident bees behave differently. The same was true for the alarm pheromone and for the oleic acid treatment. However, not for the solvent control, except for the self-grooming, but this can be easily explained with the number tag, because then they don't really like it, they always try to get rid of it. So here in our more natural setup, scented and unscented bees behaved differently. And then let's have a look at contrasting the different odors again. Now for the bees differently. So the resident bees first. We see that the antenating is significantly increased for the towards the oleic acid treated bees compared to the control, while the treated bees show no difference. They do not show more antenating towards the resident bees. So scented and unscented bees also behave differently in response to different odors. So is this setup suitable to test odor preferences? These are promising results, but they're highly weather dependent, and it's a highly uneven number of individuals, because I introduce one treated bee and I cannot control how many resident bees interact with my treated bee, so I have no control over this. So also this protocol needs to be adjusted. We think that it's better suited to perform experiments with the brood, because I can precisely control and I can choose how much cells I want to manipulate. And the setup does not currently exist because there's a problem of tracking the hygienic bees and the open cells, and to actually capture the hygienic behavior. And an update on this will also follow in a few slides. So if we now come to the continuation of the arena setup, basically the experiments are done exactly the same way, just that now everything is video recorded. Again, 20 minutes plus 5 minutes waking up time, and the tracking is done with Deep Lab Cut, which is my next topic. Deep Lab Cut, easy to use post-estimation software, where now my job was to create a suitable network, or I like to call it a never-ending story. And I didn't like it as much as this wonderful movie, where first I manually annotated frames, which were initially 800 frames from 10 videos, where I then ended up with this nice frame. What I annotated was the left antennae, the right antennae, the tip of the proboscis, middle of head, middle of thorax, and end of abdomen. And then you could see the pose of each bee, and I did that for all the bees. Here, a huge shout out goes to Lachlan, because without him, I would have not been able to do all of this. So thank you very much. Then step 2 was to create a training data set and to train the network and evaluate the network. So here we see my labels are the star ones that I manually annotated, and the round ones were done by Deep Lab Cut. And we see there it's quite good, but here there are still quite some errors. So then what had to be done was to manually correct the points made by the network. Then here you can see the machine labels, and I corrected them in the way I wanted to. And then in the end, I ended up with a quite good network with a pixel error of 4.7. Still not perfect, but this is now, after 10,000 steps, how it looks like at the moment. So this is a video that was just analyzed by Deep Lab Cut. I didn't do anything. Here you can see these points are still a little bit jumpy there, but especially for this bee, the tracking is quite good, but also here the work is still ongoing. Now let's come to the future directions and the continuation of the observation hive setup, where this winter season I will make use of the greenhouse to safely keep infected colonies and start to establish experiment where I can manipulate and track the hygienic bees. So I will introduce an observation hive into the greenhouse and do long-term video recordings where basically all bees are tagged with individual QR codes. Then the cells are manipulated, for example, by introducing varroa. The methods to track these bees does already exist and work quite well. However, right now I'm collaborating with Jacob Davidson to develop the automated tracking of the comp status, because as you have seen in the video, detecting the behavior, especially if you observe a whole frame, it's not so distinct. But what is distinct if a cell is reopened? Because I know when a cell is kept, it should be closed for X days, and then any change in the cell status tells me, oh, there must have been a hygienic event, and then I can go back and actually look exactly which bee was performing the hygienic behavior. And this is the ultimate goal, to have a semi-automated tracking of the hygienic behavior to collect the hygienic bees. Because a big drawback in the study of social immunity is that it's a collective behavior, and so what people mostly do, they introduce the manipulated cells, they wait for 24 hours, they remove the brood frame and just see, oh, 90% of the cells were removed, so it's a high hygienic score. But we have no idea which and how many bees actually clean 90 of the cells. Is it that more bees do it, or just that a few individuals do more work? We really have no idea. And with this tracking, this would allow us to test the bees that actually perform the hygienic behavior. And these bees could then be used to perform the odor preference assays, the electroantennograms, and calcium imaging. Then I'm also happy to talk about a side project that was just started, and we just got confirmed by the cluster for a small grant project, which is the Sound of Bees, a collaborative project within the cluster with Andrea Giovanni, with James Foster, Jacob Davidson, and Alberto Doimo, which is about recording the acoustic stop signals of bees. So stop signals are used by dance followers to interrupt the dancing of the focal bee. So it's really like a zzz, which tells you stop. And this can be due to predation. So a bee was following the dance, but had some horrible experience over there. Then she might perform a stop signal to say, don't go forage there, it's dangerous. But the exact meaning of why this behavior occurs remains unclear. So we want to develop an observation hive setup to record and track the acoustic signals. So introduce microphones within the observation hive. And the hive will be a collaborative use for multiple behaviors, for the dance communication, which James is really interested in, the hygienic behavior, which I'm really interested in, and for example, swarming prediction, and of course, the acoustic signals. And with that, I'm finished. I would thank you for your attention. If you have questions, you're free to ask. And of course, finally, I have to thank everyone that helped me. I guess almost every one of you have helped me, and I'm really happy to be here. And yeah, thank you all so much. And I'm happy to take questions.
Thank you, Valerie, for this beautiful overview. Amazing amount of work in the first 6 months here and a half a year. Are there any questions?
Thank you. So for the APIs experiments, did you blindfold the animals?
No.
Because I think if I remember correctly from proboscis extension experiments or learning experiments, that they learn better when they're blindfolded.
But they should not learn. So I'm not.
Yeah, exactly. But then maybe
Right.
I focus
Yeah,
more strongly on
yeah, that's a good idea, maybe. Because, right, I don't want learning, I want a natural preference to occur, but that's a good idea. Maybe we could try that out.
I'm great on getting the the difference between the treated and non-treated group at the end.
Yeah.
Yeah. I'm just wondering, do you think the treatment itself might have an effect? As in, if you perform kind of a blank control where you treat the bees without really giving any sense at all. Do you think those bees would behave differently? It would be a good blank control in your design.
We are also doing this right now, so just not giving any odor.
I see. But you still give these bees the treatment, the whole package?
No, they just get marked.
I see. No,
Yeah.
I'm saying that the treatment itself might have an effect.
Yes.
So you
Yeah.
still need to grab the bee. You still need to do all the same stuff.
Yeah.
And then put it into the colony. Mm-hmm. But then this bee is actually not treated, so it's like a blank.
Yes, we will also do that.
Okay.
Yes.
That'd be great. And then in the
But I suppose even being handled, and especially the tag, at least for the bee that is tagged, there's a difference in behavior.
yeah, and
for
that
sure.
blank control would be able to explain
Yeah.
all this weird behavior just
Yes.
coming out of the treatment.
Yeah, that's
But not
that's for sure. That's the treatment. So that the increase in self-grooming is for sure at least partly putting the tag is partly responsible for that. Yes, that's true.
And then in your earlier, the Y-arm experiment,
Yes.
right? Have you considered some reward or feedback? I don't know about bees that much, but usually if they are attracted to odors, right? Do they get any feedback, like when they perform a behavior, right? Because in your experiment, there's no feedback.
Yes.
There's no.
Yes.
So maybe that's
So
why you're not getting the differences.
Yes. So I want to have so to say, the innate preference of the bee. That's why I'm not giving any reward. But it's true that, of course, the reward, like of odor, for the bee is associated with an appetitive stimulus, at least the floral odor, right? The bee kind of expect to, if I go there where it smells good, I should get a sucrose reward. But I try to get an innate preference for the odor without giving anything, because then it's learning, and then we do not only have the point of which preference has the bee, but also how well does the bee learn, which again introduces variability. But yes, we are now, we were with the workshop, we did an extension of the Y-APIS setup where we now introduce an artificial flower to also give sugar reward inside the Y-APIS, exactly for what you were saying. And we are in the trial and error phase of getting the new setup to work. So this is definitely something that I will be looking into the future. Yes.
Yeah, I'm just wondering, because from the perspective
Yes.
of the humans, right? If
No,
we find
but
something like right? If we try to get it and there's nothing at all, right?
Yes.
We might get upset and all that.
Exactly.
That might kind of
Exactly. Yes.
cancel out of behavior.
Yes, so we will definitely try this, because I mean the bee maybe goes to the odor but then quickly learns, I'm not getting anything, so why should I go there? Yes.
Thanks a lot. Great talk.
So this last comment could explain why in your data on the first day you find an effect and then the second day
Yes.
you do not find an effect.
Yeah.
Because it gets a habituation to,
Yes.
to the situation.
Exactly.
Any more questions?
Well, just a generic comment related to this idea of habituation. I don't know if you consider it. Usually animals have this ability after a while to be exposed to some odors to not feel it anymore.
Yes.
You think is playing somewhere a role?
Yes.
Try to control.
Could very well be. Especially we don't really know how well our vacuum pump works. And so it could be that even after 2-3 trials, it just smells like a big mix of everything everywhere. And then the bee might just get used to it, yeah, for sure.
On the other side, in a very infected hive, there should be quite a lot of this
Yes.
odor, and they shouldn't habituate, because otherwise they don't act. So I have no idea.
Yeah.
I'm just throwing.
Or at least different bees should habituate at a different rate.
Exactly.
So I would
right.
expect that the hygienic bees should habituate much slower to the disease odorant, while maybe a forager habituates quite fast, because for the forager bee, the disease odor is not as important as, for example, a floral odor. And also the sensitivity, how sensitive you are to stimuli really depends on the age of the bee and the task she's performing.
And if there is a difference between the individuals, you are back to your question that you had
Yeah.
in your PhD. What is the individuality, and how does the difference in behavior from one individual to the other one contribute to the entire hive and the collective? I don't see any more questions, in which case I would like to thank you. And thank you all. And we'll be back for the seminar next week.